Mutation are of two types: point mutation and frameshift mutation. Point mutation deals with base substitution, which can be of two types: transition and transversion. Frameshift mutation is related to addition or deletion of a base. In transition, a purine base is replaced by a purine base, or a pyrimidine base is replaced by a pyrimidine base. In transversion, a purine base is replaced by a pyrimidine base, or vice versa. Now, before we get into the details, we'll see some basics of DNA, which says purine bases are adenine and guanine, both in DNA and RNA. Pyrimidine bases are thymine in DNA, but in place of thymine, in RNA, uracil is present. And cytosine is another pyrimidine base, which is present in both DNA and RNA. Now, we'll see some examples related to transition and transversion. Now, before we get into the examples, we'll see that the central dogma suggests that from a DNA, mRNA is formed, and from mRNA, amino acids are formed that will result in protein formation. Now, get into the example. A thymine base is replaced or substituted by a cytosine base. That means both are pyrimidine bases. So a pyrimidine base is substituted by a pyrimidine base. So this will come under the category of transition. Now, see another example which comes under transversion is a thymine base is replaced by a guanine base. So a pyrimidine base is substituted by a purine base. So this will come under the category of transversion. Let's discuss the effects of point mutation. It can be of three types: silent, missense, nonsense. We'll try to understand this with the help of related examples. In silent mutation, a change in base from cytosine to thymine will not result in any change in the amino acid. The amino acid will remain same. This occurs due to the degeneracy of code and mainly when the third base is changed. Now, what is degeneracy of code? We can see that a number of codes are coding for an amino acid, that is leucine, and two different codes are coding for an amino acid, for example lysine. Now, in every codes we can see that only the third base is changed, and the change in third base is not altering the amino acid. We will see one example of nonsense mutation, when a change in base may result in a stop codon. Over here, a change from thymine to adenine base is resulting in a stop codon, that is UAG in mRNA, that will not code for any amino acid and will result in premature termination of a polypeptide chain, making it non-functional. What are the stop codons? UAA, UAG, and UGA. In missense mutation, a change in base results in change in amino acid. In this example, we'll see a change in base from thymine to cytosine result in change in amino acid from lysine to arginine. Missense mutation can be further divided into two types: conservative and non-conservative missense mutation. We'll see some examples related to conservative and non-conservative missense mutation. In conservative missense mutation, we'll see that a change in base from thymine to cytosine will result in change in amino acid from lysine to arginine. But in this case, both lysine and arginine have similar properties. So in conservative missense mutation, we'll see substituted amino acid has similar properties compared to the original amino acid. In non-conservative missense mutation, we'll see a change in base from thymine to guanine will result in change in amino acid from lysine to threonine. But both lysine and threonine have different properties. So in non-conservative missense mutation, substituted amino acid has different properties compared to the original amino acid. Let's discuss some medical effects of point mutation. In silent mutation, since the the amino acid is not changed due to the change in base, there will be no significant or disastrous medical effect. In missense mutation, the medical effects can be categorized as acceptable, partially acceptable, and non-acceptable. We can see some examples under these categories: hemoglobin Hikari, hemoglobin sickle cell anemia, and hemoglobin methemoglobin. In all these examples, due to change in amino acid, hemoglobin is affected, which may alter its oxygen carrying capacity. For example, hemoglobin Hikari, lysine is replaced by asparagine at position 61 in the beta chain. The mutant hemoglobin is capable of normal functioning, so it will come under the acceptable category. In hemoglobin sickle cell anemia, glutamic acid is replaced by valine at position 6 of beta chain. The hemoglobin S is an abnormal form of hemoglobin that causes RBCs to become rigid and sickle shaped. The rigid misshapen blood cells do not carry oxygen well, and it partially affects the oxygen carrying capacity. So it comes under the partial acceptable category. In non-acceptable category, hemoglobin M, histidine is replaced by tyrosine at position 58 of alpha chain. This makes hemoglobin M incapable of combining with oxygen, so it will come under the non-acceptable category. Let's see an example under nonsense mutation, beta thalassemia. It is a hemolytic anemia due to deficient synthesis of beta globin chain. It may occur due to nonsense mutation or frameshift mutation that will block translation and prevent beta globin synthesis. You see a code AAG coding for lysine in the beta chain. Suppose at position 17, adenine is replaced by uracil. It will generate a termination or a stop codon that will not code for any amino acid and thereby prevent beta globin synthesis. You try to understand point and frameshift mutation with another example. The sentence on screen says, "The rat saw the man." Now suppose the letter R is substituted by the letter C. The sentence becomes, "The cat saw the man." There is a slight change in the meaning of the sentence, but the entire reading frame is not disrupted. Therefore, in point mutation, we can say that a substitution of a base will not disrupt the entire reading frame. Over here, we can see only one word is changed from rat to cat. Rest of the words remain same. We can see point mutation with the help of an example. The DNA bases are as such. The corresponding mRNA bases are as follows, which will code for particular amino acids like histidine. If there is a substitution, say G is replaced by C in DNA, the corresponding complementary base in mRNA will also change, which will cause a change in amino acid as well. But in case of substitution or point mutation, we can see that only one amino acid is altered. Rest of the amino acids remain same. We use the same sentence to understand frameshift mutation, where mutation is caused by addition or deletion of base. The sentence on screen reads, "The rat saw the man." If a letter C is inserted between the and rat, and we consider 3 letters in a word, signifying a genetic code which also exhibits 3 bases in a code, then we see not only one word but the entire reading frame is disrupted and there is a shifting in the entire reading frame. Now, not only one word, that is cat, is disrupted, but the entire reading frame gets disrupted in frameshift mutation. We will see another day-to-day example to understand frameshift mutation. Suppose people are standing in a queue. A person tries to adjust himself somewhere in between the queue. The people standing in front of him will move a bit ahead to give him some space. That is, they will shift from their respective position to a new position. And therefore, there will be shifting in the entire line. Now let's try to understand the effects of insertion and deletion in frameshift mutation. The DNA bases are as such. The corresponding complementary mRNA bases are as such, which will code for a particular amino acid, say histidine. Now let's see an example of insertion. Insertion of a base, say adenine, and then considering only 3 bases that signify a genetic code, we'll see that the entire code changes in the DNA, which will result in corresponding change in codons of mRNA, and subsequently the amino acids will also change and new amino acids will be incorporated. So an addition of a base will result in shifting of the codes and changing in the amino acid. Similarly, we'll see an example of deletion, where deletion of a base A, adenine, and then considering 3 bases per codon, it will result in shifting of the codes in the DNA. Due to the alteration in the codes in DNA, the codes in mRNA also alters, resulting in change in amino acids. Therefore, we see that addition or deletion may cause shifting and disruption of the entire reading frame. Let's discuss some diseases related to frameshift mutation. Cystic fibrosis, Tay-Sachs disease, hemophilia A and B, DMD. In cystic fibrosis, mutation in the CFTR gene will result in non-functional CFTR protein that will affect the chloride channels in the cells and cause sticky mucus in various organs. In Tay-Sachs disease, mutation in the hexa gene will cause deficiency of an enzyme called hexosaminidase A and will lead to neurological problems. In hemophilia A and B, mutation in the gene F8 and F9 will disrupt the production and function of clotting factors and disrupt the clotting mechanism. In DMD, mutation in the DMD gene will cause non-functional dystrophin protein and result in muscle weakness and degeneration. Let's discuss some MCQs related to point and frameshift mutation. What is the substitution of a purine base with a pyrimidine base known as? It will be transversion. What is the substitution of a purine base with a purine base known as? It will be transition. DMD disease is due to non-functional dystrophin protein. Point mutation involves, the answer will be base substitution. In sickle cell disease, the amino acid that is replaced is glutamate is replaced by valine. Which type of mutation results in sickle cell disease? It is non-conservative missense mutation because glutamate and valine will not be related to each other in terms of properties. Frameshift mutation generally introduces a new section of amino acids. A mutation in which only one nucleotide is altered is called a point mutation. Due to base substitution, a nucleotide will be altered, then it will be related to point mutation. A nonsense mutation is one that creates a premature stop codon. If adenine is replaced by cytosine, it is known as transversion because adenine is a purine base and cytosine is a pyrimidine base. A non-conservative mutation is one in which new amino acid has different biochemical properties than the original. A silent mutation is highly unlikely to affect protein because multiple codons can code for the same amino acid. This is known as degeneracy of the code. If your DNA went from GAT, CGT, ATC to GTT, CGT, ATC, what type of mutation do you have? Now in this case, adenine is replaced by thymine. So there is a substitution involved. So this will come under point mutation. Now what type of point mutation it will be? it will be a missense mutation, because it cannot be a nonsense mutation because it is not leading to a stop codon, and it cannot be a frameshift mutation because there is no insertion or deletion of any base. Which of the following represents a transition? The answer will be G to A, because G is a purine base and A is also a purine base. So purine to purine will come under the category of transition. The type of mutation where lysine is substituted by arginine. It will be a missense mutation because in missense mutation the amino acids are changed. Insertion or deletion of a nucleotide sequence may lead to what type of mutation? The answer will be frameshift mutation. The type of mutation where the third base is changed, resulting in no alteration in the amino acid. The answer will be silent mutation.